an idea to introduce the technique, how does it work, and give a few examples of achievements that have been made by this technique. The earlier speaker, Prashant, I believe, he gave very nice coverage of the modern techniques in plant breeding, innovative techniques in plant breeding, including gene editing, and he highlighted some recent contributions of gene editing to plant breeding. Next one, please. Next. To begin with, let me define gene editing. It is a technique that induces the desired genetic change in the targeted genomic site, or make it simpler, it is a technique that induces the specified or the desired base sequence change in the gene that the experimenter selects for making mutations that would create a phenotype of his interest. Basically, three types of tools are used for this purpose. One is side-directed nucleases. As Prasant said, there are three zinc finger nuclease, ZFN. That was the first one, and then followed by two others. I'll come to that. So side-directed nuclease is one category of tools. Chimeric oligonucleotides is another category of tool. And then base editors are the third category. Next one, please. Gene editing produces genetic variation or changes in genes, just as any mutagenesis program does. But as compared to the mutagenesis program, gene editing offers certain advantages. If you induce a mutagenic agent like a chemical or a physical mutagenic agent, then the mutations are induced in random selection of genes. A large number of changes often are induced. According to one estimate, several thousand nucleotides are changed in a single plant treated with EMS. And the frequency of mutation in the gene that you are interested in is very low, 1 in 1,000 essentially. So here, with gene editing, you have almost complete control on the gene that will be mutated. You have also a control on the type of mutation that will be introduced into the selected gene. As a matter of fact, you can specify mutational changes by providing a donor DNA along with the side-directed nuclease. And the frequency of the mutation that you are interested in is very high. Typically, it's between 3 to 10%, and sometimes it can be very high, exceeding 20% or more. Next slide, please. Next slide. So the first category of genome editing tools is side-directed nucleases. As I said, base editors. Side-directed nucleases, they are called SDN for short. It started with zinc finger nuclease that was created in 1994, followed by TALENs in 2011. And then in 2013, CRISPR-Cas9 was developed. Each of these SDNs, JDF1 is the most problematic to use. TALENs are much easier, and CRISPR-Cas9 is the easiest of the three SDNs to use. Therefore, CRISPR-Cas9 has been adapted to develop tools, base editors that chemically convert single bases to another base, like they can convert cytosine to thymine and adenine to inosine or guanine. Chimeric oligonucleotides have been long in use, but they are not very frequently used for gene editing. Next one, please. Next slide. Next slide, please. Um, um, no, previous one. The site-directed nucleases have a simple design. They have two functional parts. One part recognizes the DNA sequence that you want to introduce changes at, and the other part is nuclease domain that cuts the DNA, producing a double-strand break. Next one, please. And this is the three types of nucleases. The first two nucleases are based on eukaryotic transcription factors. Zinc finger nuclease is based on zinc finger protein. This is the zinc finger protein that recognizes the DNA that you want it to bind to. And then it has a nuclease domain from FOC1 bacterial nuclease that cuts the DNA. In this case, the transcription factor is from Xanthomonas bacterium. It's called tail protein, transcription activator like element. Then this DNA binding area is attached to FOC1 again, that cuts the DNA. Both Zediafian and tail talent, they have to be used in pairs. One cuts one DNA strand, the other cuts the other DNA strand to produce double strand break. And at the same time, their designing involves modification of properties of these proteins. And protein engineering is highly sophisticated, very demanding, and requires lots of chemical expertise, which is not usually available with many biological laboratories, actually any biological laboratory that would be interested in gene editing. So it was great relief when CRISPR-Cas9 came along. The CRISPR has two components. One is RNA, and the other is Cas9. That's protein. This is guide RNA. This is Cas9 protein. Next one, to make it simpler, the guide RNA has two parts. One is the pairing region. This differs from one CRISPR-Cas9 to the other because this is the part that recognizes target DNA sequence and pairs with by complementary base pairing. So this 20 nucleotide long region is the only region that the experimenter has to redesign for a new target. This is the backbone region or scaffold region that associates with Cas9 and activates Cas9 to do the nuclease, to function as nuclease. And the Cas9 nuclease has dual purpose to first associate with target DNA. So it associates with the DNA molecule at a specific site and then begins to unwind the DNA. DNA unwinding will continue if pairing region is homologous to the region following the site where unwinding begins of the target DNA. If it is not complementary, Cas9 will dissociate and move on to another site. So what it does is, next one please, next slide. The guide RNA pairs with the target DNA. Next one, sorry, previous one. Guide RNA pairing region pairs with the target genomic DNA. This is genomic DNA. And Cas9 is there, present there. Both Cas9 and guide RNA must be associated to be able to do this pairing. There is unwinding of the DNA, and there is induction of double strand break at around here. Next one, please. So what the site-directed nucleases do is to induce double-strand breaks. They don't induce mutation. Mutation will be produced by repair of the double-strand break by cellular machinery for DNA repair. And that is how genetic changes are generated. This DNA repair, next one please, is of two types. Uh, the most of the double strand breaks will be repaired by what is called non-homologous end joining. In this case, the two broken ends of the DNA are brought together and joined by DNA repair enzymes. Generally, this end joining is very precise and there is no change in base sequence. But in low frequency, there will be either addition of a base or there will be deletion of a base. And since the SDN is always present in the cell, where once repair joins precisely the cut ends, the nucleus will cut it again. And this will cutting, joining will go on till such a time when there is insertion or deletion of bases so that the nucleus no longer recognizes this cut site. That is why there is so many double strand breaks produced at the targeted site. When only SDN is used, this is the kind of repair that will happen. And the repair of DSB will be generating insertion and deletion. And as a result, there will be inactivation of the gene that you target. These changes are referred to as SDN1 events, site-directed nuclease 1 events. And they are the simplest to induce, and they are exempt from any regulation. They are not treated as transgenic, provided there is no foreign DNA present in the gene-edited product. Next one, please. To summarize this, how non-homologous end joining works, this is the genomic DNA. The site-directed nuclease cuts it, producing a double-strand break. There is non-homologous end joining. Ultimately, this repair process leads to either deletion, so that some bases are lost, or there is insertion of few bases. In either case, insertion or deletion of the bases will lead to gene inactivation by this, that if there is one base added, two base added, the reading frame of the base sequence will change, and therefore there will be wholesale amino acid replacement in the protein, and that will become non-functional. But if it is addition of 3 or deletion of 3 or multiple of 3, then change may not be that strong unless a stop codon is created. So that kind of mutational change will also take place, but that's frequency of such mutational changes is low. That is why we talk about insertion deletion, because they are bulk of the activity and the result is gene inactivation. Next one, please. At this point, let me briefly outline the procedure for gene editing. And the first thing required for gene editing is that you know the gene that you want to change. Okay, so the gene you want to produce a phenotype. Say, for example, this is tomato. If you are familiar with tomato flower, this is the flower. This is the pedicel that joins to the stem, and this pedicel has a joint. So Tomato fruit will be borne on a sharp branch that has joint and then attached to the stem. So if machine harvester is used, it will pull the fruit from the tomato plant. As a consequence, along with the fruit will come out the sharp piece of pedicel and the calyx that is associated with the fruit. Uh, in US, tomato is mechanically picked, and after picking, this pedicel and calyx has to be manually removed. That's very costly. So what you want is to produce a tomato flower that does not have that jointless pedicel. Okay. So you know the phenotype that you want to produce, and now you need to know what is the gene that is responsible for this. Genetic studies have shown that it is the J2 gene that produces jointless pedicel in tomato. So J2 gene was identified, sequenced, and then it was determined what kind of base change will produce jointless pedicel. The conclusion was that any base change that makes this gene non-functional will produce a jointless pedicel mutant. So what was done? Normal tomato was, so once you have this knowledge, you want to design a side-directed nuclease that will attack gene J2. So you design that nuclease, you introduce that side-directed nuclease into cultured tomato plant cells and let um, grow for one or two subcultures, then regenerate plants from these transformed cells. You can select for cells that were transformed by the side-directed nuclease introduction. And from you can introduce plants, regenerate plants from them, grow them, and evaluate the flowers that they produce, either in the same generation or maybe in the next generation. And because a phenotypic evaluation in T1 generation, that is the generation produced by selfing of the T0 generation, you will be able to identify some mutants with this kind of flower that has no joints in the pedicel. Um, you can confirm the mutational change by PCR amplification of this gene, sequencing it to identify that there has been sequence change in the J2 gene of the tomato. Next one, please. So in simple terms, you decide on the phenotype, identify the gene, create SDN that is specific for that gene, introduce that SDN into cultured plant cells, let it carry out gene editing, regenerate plants and evaluate the plants either by some molecular technique or by phenotyping to identify the mutant that you are interested in. In plants, there is a fungal pathogen, many plants, including wheat, powdery mildew. The powdery mildew fungus attacks plants with the help of a protein that is called MLO protein. MLO protein is produced by the host plant and is necessary for some transport function in the plants. But the fungal pathogen uses that MLO function to enter into the plant cells. If you inactivate MLO gene, then fungus is not able to attack the host plant. Wheat has three MLO genes, one on A genome, the other on B genome, and the third on D genome. And at least two of these, are preferably all the three of these, have to be inactivated at the same simultaneously to create resistance. Obviously, simultaneous inactivation of all the three genes in the same plant is nearly impossible to achieve by any mutagenesis technique. But with gene editing, this has been achieved. What was done that a sequence that is conserved in all the three homologs of MLO gene, that sequence was targeted so that the SDN will bind to all the three conserved sequence, the conserved sequence in all the three genes, uh, create DSB and inactivate the gene. This has been achieved in many other plants. Next one, please. Okay. Then we come to what we call site-directed nuclease. Two events are SDN, two events. Um, these events involve homology-directed repair, in which, in this kind of repair, the double-strand break is repaired by using a DNA as template that is homologous to the genomic region where the break has occurred. So what experimenter does is he provides the donor DNA along with the site-directed nuclease, introduces into the cells. So donor DNA is there, site-directed nuclease is there, and when double-strand break is induced, the donor DNA will be used in some cases at least for homology-directed repair. The donor DNA is a DNA sequence that is identical with the genomic region where break has occurred on either side. So the donor DNA has a sequence that is present on one side of the break and also on the other side of break. In the center of this donor DNA, however, the experimenter puts in the base sequence that he wants to introduce into the gene or genome, and that way create mutation. So during the pair, the donor DNA sequence is copied into the genome, and this sequence copying or replacement will either correct a mutation or induce a new mutation. It can also lead to integration of complete genes at the DSP site. And I'm sorry, there is some background noise. Uh, can you hear me? Should I continue? So the Maunam Srukit Lakshanam, I'll continue. Sequence replacement will either correct a mutation or it will create a new mutation as desired by the experimenter. The experimenter can use homology directed repair to introduce a complete new gene construct at the targeted genomic site. When you introduce complete gene, this is called SDN3 event. When it is a small sequence that is introduced into the gene, this is referred to as SDN2 event. As Prasant said, SDN1 and SDN2 events are exempt from regulatory oversight, provided these products do not carry any foreign DNA, and that has to be demonstrated by specific tests. Next one, please. Uh, to give you pictorial representation of this homology directed repair, this is genomic DNA. This is where the double strand break has been introduced. The donor DNA that is provided, this part of donor DNA is homologous to this part of the genome. This part of homologous donor DNA is homologous to this part of the genome. And this is the new sequence that you want to introduce at the site of the break. This may be few bases, this may be a larger base sequence. And in that case, the base sequence, the genomic region here, uh, that is uh, not included in the homology region, that will be replaced by this sequence. So after repair, this sequence that is different is copied into the genome, and the sequence replacement takes place. Uh, like if crossover or recombination occurs here and recombination occurs here, homologous parts of donor DNA will also be included, but that does not matter because this base sequence is the same as the base sequence here. Only this one matters. So that is what is the replaced sequence shown here. Next one, please. An example, successful example of SDN2 technology is that maize is affected by a disease called northern corn leaf blight. This resistance is found in some inbred lines of maize, but transferring the gene from a resistant inbred line into a susceptible inbred line will take about 6 to 7 years and considerable amount of phenotypic testing. So what was done that uh, donor DNA was used for homology-directed repair. This donor DNA has the sequence that is present in the resistance allele and responsible for resistance. This sequence, this donor DNA was used along with the SDN to produce a genome edited line that was resistant to northern corn blight. And this line, gene edited line, has been approved by APHIS, USDA for commercial cultivation. The advantage of this is that this approach is that you can, once you know the sequence that is responsible for production of the desirable trait, that sequence can be introduced into elite inbred lines or varieties within a period of 2 to 3 years and without going through all the back crossbreeding efforts. So that reduces the time and the effort required for creating the desirable genotype in an otherwise elite variety or inbred line. Next one, please. SDN3 events, as I mentioned to you, is integration of complete gene at the specified genomic site. And what is done is that DSB is into double strand break is introduced at the genomic site, the specified genomic site. And the donor DNA used has the complete transgene construct. So homology directed repair will introduce the gene, transgene at that site. Um, in normal transgenic protocols, the genes are integrated at random genomic sites, which leads to which may lead to either inactivation of desirable genes or activation of undesirable genes. And often this also exposes the transgene for transgene silencing. But by choosing the genomic site where the transgene will be integrated by gene editing tool, you avoid the risk of silencing of the transgene. You also avoid the risk of inactivation of desirable gene and activation of undesirable gene. So that makes the transgene product, transgenic product, much safer. Excuse me. And hopefully, this would minimize the biosafety tests required by the regulatory authorities for clearing of transgenics produced in this manner. Next one, please. There are some issues that are associated with gene editing that is referred to. One of them is off-target cleavage, which means that the DNA cleavage occurs at sites that are similar to the targeted site. In addition to the targeted site itself, the occurrence of off-target editing has been minimized by genetic engineering of the site-directed nucleases. But this cannot be possibly eliminated totally because The systems that we use for gene editing, like CRISPR or zinc finger or tail protein, they have a evolutionary history, and it is obligatory on them to maintain some kind of non-fidelity, that is promiscuity, that is some kind of leeway in pairing between the SDN tool and the DNA. Uh, the other problem is predominance of non-homologous end joining. Efforts are being to minimize this, and some tools like prime-based editors have been created that show uh, much less non-homologous end joining that normally. But unfortunately, even prime editor does not enhance the frequency of HDR product. It only increases the proportion of HDR among the total gene edited products. And the frequency of homology directed repair is low. Um, this this is a great problem when you come to apply gene editing to human subjects for treatment of diseases and so on. And then the last difficulty is that the donor DNA plasmid may integrate into the at the target site, which is highly undesirable. And this can be avoided by using the gene editing tools like JDFN, TLN as proteins, and CRISPR-Cas9 as ribonucleoprotein, the RNA, sgRNA, and protein Cas9 as prepared, not as DNA, introduced into the cell, not as DNA, but as ribonucleoprotein or protein only, so that there is no risk of DNA integration. Uh, these techniques are being refined, and hopefully, in due course of time, they might become routine. But at the moment, to use proteins and ribonucleoproteins, you need transcription, in vitro translation, and such facilities, which may not be available to many workers. Next one, please. Next one, please. Uh, this is chimeric oligonucleotide that has been used for mutagenesis. And these were the products from this mutagenesis were the products that were initially approved in Europe, but now it appears that Europe is going to accept other gene editing products from CRISPR-Cas9 tool also. A chimeric oligonucleotide is a oligonucleotide that has hairpin-like structure, and in the center of this whole part is DNA. In the center of it, it has short regions, 10 bases on either side, which are RNA. These black strands are DNA. The red one is RNA. So you have, and in the center is 5 base region, that is DNA. The RNA here is used to provide higher efficiency in base edit, or sorry, in gene editing due to oligonucleotide. Um, in the DNA sequence that is in the center, the red ones are the ones that are different from the genomic region. So what you do, this base sequence is designed by the experimenter and changed from the base sequence that is present in the genome. Rest of the oligonucleotide is complementary to the target genomic region. So this is genomic DNA. The oligonucleotide will become single-stranded and base pair with the 2 strands of DNA, genomic DNA. And the mutation carrying regions, or the base sequence change regions, will also be present with the genomic DNA strand. Um, sorry. So the when next time DNA replication occurs, before that there will be excision repair. There will be a mismatch. This base sequence is different from base sequence present in the genomic DNA, and therefore there will be mismatch and there will be a bulge. This mismatch will be repaired by DNA repair machinery called excision repair. On an average, half of the time, the mutated bases or changed bases will be deleted, but half of the time, the genomic region, bases in the genomic sequence will be deleted, and this will be used as template for repair. So at least 50% of the time, the base sequence will change because of the base sequence changes that were included in the chimeric oligonucleotide. And this leads to generally base change of or base replacement of 1 to 3 bases, but even 5 bases in some cases have been replaced by this technique. This can also be used to induce insertion and deletion of few bases, 1 to 3 bases. And in the end, you will be able to isolate mutants that carry the mutational change that you had induced. Next one, please. Next one, please. Ah, thank you. Then finally, we all know that DNA after replication and the chromatin um, after formation of the uh, chromatin fiber undergoes certain changes that are temporary. These changes are added to DNA and histone, and they are removed from DNA and histone. So therefore, they are referred to as epigenetic changes, and and tools have been created to induce these epigenetic changes, and they are called epigenome editors, and the process is known as epigenome editing. Um, for creating epigenome editors, what is done is that you don't use nuclease Cas9. You kill the nuclease activity of Cas9 by mutation, so that it is not no longer able to cut DNA strands. So this is referred to as dead Cas9 because Cas9 activity is cutting DNA. If it cannot cut, then Cas9 is inactive, and that is called dead dCas9. To Cas9 is fused either transcription activator domain or transcription suppressor domain. A transcription activator domain will remove, let's say, uh, DNA methyl groups from DNA, or it will acetylate histones. The suppressor, on the other hand, will induce DNA methylation or will induce deacetylation of histones, and thereby a transcription activator will activate transcription of the gene that you target. A suppressor will suppress transcription of the targeted gene. What is done is the epigenome editor or activator or suppressor, that is Cas9 carrying activator or suppressor domain, is associated with this guide RNA that targets the promoter region of the gene that you want to activate or suppress. And a single activator or suppressor will not be enough. You need multiple activators or suppressors to produce notable suppression of gene transcription or to induce sufficient level of gene transcription. And this has usefulness in treatment of human diseases like such as HIV. Possibility is there. Last I followed up was that a technique for uh, treatment of HIV based on transcription activation was being developed for clinical trial. Whether it has reached there, I have no idea. I have also, I hope that this technique will find uses, applications in plants. It has been shown to act in plants, and in at least one case, that is resistance to drought, this has been shown to be effective in inducing drought resistance in plant by activating a drought resistance gene using transcription activator DCAS9. Next one, please. Then we come to base editing. Base editing is permanent conversion of a base into another by chemical modification inside the cell itself. This chemical conversion can be easily done outside in test tubes, but in case of base editing, it is done within cells, either using a kind of base editor. These base editors are basically two types, excuse me, adenine base editor and cytosine base editor. The CRISPR Cas9, referred to as N-Cas9 or Cas9 nickase, that is used with deaminases um, to create a base editor. A nickase Cas9 is a Cas9 that is able to cut only one DNA strand instead of both DNA strands. Normal Cas9 cuts both the DNA strands and induces double strand break. But a nickase Cas9 only induces a nick at the targeted site. The base editors report came first in 2016 by Komor and co-workers. Next one, please. The architecture or design of base editors is like this. This is nickase Cas9 in black, and this is site adenine deaminase, denoted by TAD A by red. This adenine deaminase is called TAD because this is a tRNA adenine deaminase that was naturally uh, functional on tRNA adenines only. It did not edit adenines present in DNA molecule. This protein was evolved in vitro by what is referred to as directed protein evolution technique to create an adenine deaminase that would function on DNA instead of tRNA. That is why it is shown as star here, that it is genetically engineered tRNA adenine deaminase. So when you use N-Cas9 along with this gRNA, the Cas9 and guide RNA will seek out the target site and associate with it. It will also cause unwinding of the DNA and create a window where this adenine deaminase will find adenine and deaminate it to create ultimately A to G transition. A cytosine base editor is created by fusing a cytosine deaminase. One such enzyme is R-ApoBEC1, and this is fused to N-Cas9, and then uridine glycosylase inhibitor enzyme is associated on the C-terminal end. This is done to prevent excision of uracil because we will see that later. So this is how these two base editors are designed. Next one, please. When cytosine is deaminated, it produces uracil. Normally in the cell, uracil is removed by uridine glycosylase enzyme, and there is excision repair. So U is replaced by C. But when excision repair is blocked by using UGI, then uridine remains in place of C in the DNA strand. When DNA replication occurs, in place of U, there will be T incorporated. So there is C to T transition because of C to T base editing. Next one, please. Next one, please. An example of application of C to T base editing is to create herbicide resistance by editing the gene that encodes acetolactate synthase. This herbicide inhibits acetolactate synthase. And to create acetolactate synthase that is resistant to the herbicide, C at position 287 was converted to T, as a result of which alanine was replaced in the protein by valine, and thereby the acetolactate synthase enzyme became resistant to inhibition by the herbicide. This was done in rice, and the rice plants produced by gene editing, they were resistant to the herbicide. Next one, please. In A to G base editing, A is deaminated, and because of deamination of adenine, inosine is produced, inosine. For all practical purposes in the cell, I functions as G during DNA replication, during transcription, during translation. So conversion of A to I is essentially equivalent to conversion of A to G, but I is not normally present in DNA. Therefore, when DNA replication follows base editing, I is replaced by G, so that there is transition of A to G. Next one, please. Uh, I may add here that techniques are being developed. Not only we can induce only A to G and C to T transitions, but by targeting bases in the complementary DNA strand, you can create all four types of transitions. That is, G to A and T to C also can be targeted, can be achieved. But transversions, that is A replaced by G, C, and T, G replaced by C, T, and so on. Transversions are not normally produced by base editors, but some base editors are being developed that can produce substantial amounts of transversions. That is replacement of an adenine, a pyrimidine by a purine, and a purine by a pyrimidine, and so on. What you can achieve by base editing is there can be correction of a mutant gene to a normal wild type gene, and this will be useful in genetic disease correction, particularly when they are produced by GC to AT transition. The base editing can be used to correct it. To induce a desired mutation by single base substitution. It can also be used to inactivate, theoretically at least, to inactivate the targeted gene by producing a premature stop codon. But if inactivation is desired, then site-directed nuclease 1 strategy is simpler and much preferred. The base editing now is being used for directed endogenous protein evolution. To do gene editing, as I said, you need to know the gene, you need to know its sequence, you need to know the base sequence that has to be changed to produce the desired phenotype, and also the kind of base sequence that has to be brought in to produce the desired. So there has to be a detailed knowledge of relationship between gene sequence and its function in phenotype production. But in protein evolution, you need only two information: the gene that is responsible for a phenotype and sequence of this gene. You need another thing is a test for the function that you want to achieve after editing. So you should be able to select for the desired function after the protein has been subjected to editing. So what you do is in protein evolution is you produce all kinds of base changes, base sequence changes randomly. Millions and millions different types of base sequences produced of the same gene, and then regenerate plants or select cells for the. Suppose you are using evolving a gene um, that that is involved in herbicide resistance, like ALS, acetolactate synthase gene that is involved in herbicide resistance. You want to produce new alleles of this gene. So what you will do, you will subject it to all kinds of base sequence changes and select for herbicide resistance. The base sequences that will enhance herbicide resistance will be selected, and the plants regenerated from them will show enhanced resistance to the herbicide. Um, Similarly, other traits could be selected for. There are examples of herbicide resistance selection by protein evolution, but there are 8 examples to emerge for use of this technology for production of improved quality or improved yield or some other characteristic. Next one, please. Uh, there is this is a very unusual application of site-directed nucleases. This is to create gene drives. Gene drives are actually genetic devices that are capable of spreading in any crossbreeding population once they are introduced into crossbreeding population, so that ultimately they invade all the chromosomes, all the homologous chromosomes present in the crossbreeding population. The gene editing, the gene drive is essentially a donor DNA. This homolog H1 is, you remember the donor DNA that at the site of double-strand break, on left side, there was a sequence that was homologous to the genomic sequence. This is homology H1 region. Similarly, homologous sequence to the genomic region is H2 region. In between these two, there is Cas9, guide RNA, and any gene that you can put in, any gene that you want to introduce into the population. That is referred to as payload gene. are a gene that is carried by the gene drive. This guide RNA is specific for the genomic sequence where the which has this H1 and H2 genomic sequences. So it will produce double strand break. It will pair there, Cas9 will cut, and homology directed repair will copy the gene drive into the uh, chromosome region. Next one, please. Uh, what what is the use of a gene drive? It can be used to change the genetic makeup of a crossbred population, like composite varieties of maize, say open pollinated varieties of maize. You can introduce a gene of interest there easily and quickly. It can be used to rescue a threatened population uh, by using pollinator insect species. It can be used for eradication or suppression of a population like weeds, pests, and mosquitoes. In this context, I should point out that a British company, Oxitec, is testing a gene drive for control of mosquitoes in Brazil. They have not yet published the result. Some gene drives were being tested, evaluated in USA for control of plant pests, but those results are still not at least available to me. They might have come in literature. Next one, please. Um, uh, this is the gene drive that is being tested by the Oxitec friendly TM mosquito platform. This is what they call it, and this is being used to control Aedes aegypti. The area where it is being done is Brazil, and this gene drive causes female sterility so that the female mosquitoes are unable to produce progeny, and there is check on the spread of the gene drives. Next one, please. As I said, gene editing, there are certain prerequisites. The critical gene that needs to be edited, that must be identified. The target site within this gene that needs to be mutated, that must be determined. And the base sequence that is to be changed at that site, that must be decided on the basis of available evidence, so that you get, after the base change, the desired phenotype. CRISPR-Cas9 designing for editing any gene is simple, as I said, that you only need to redesign the 20 nucleotides of a pairing region of guide RNA. And then gene editing protocol has to be implemented. That is, after you have created the gene editing tool, you transform it into cultured plant cells, select for transformed cells, regenerate plantlets from the transformed cells, and evaluate the progeny of the regenerated plants for the gene editing mutational changes, either by phenotypic evaluation, by sequencing of DNA, or some other molecular technique. Next one, please. Uh, a futuristic and attractive to human beings application of gene editing. Excuse me. um, in blastocyst stage embryos of animals, um, if you are an animal scientist and I make an error in using a term, please excuse me. I'm a plant scientist, but this attracts my imagination, so I'm trying to share it with you. At blastocyst stage of animal embryo, Fates of specific cells and cell groups are already decided that this cell group will give rise to a particular particular organ of human body or animal body. So, for example, there are cells that are predetermined at blastocyst stage to develop into pancreas. Okay, so what is done is that gene editing is used to inactivate the gene that leads to formation of pancreas. The gene that leads to formation of pancreas that is inactivated so that the blastocyst does not have cells that will develop into a pancreas. What is done is, if animals were developed from this blastocyst stage embryo, they will be deficient in pancreas. Whether they will survive, and I don't think they will be able to survive. Human differentiated cells are, let's say, human skin cells or any tissue culture cells that are taken from primary explant. They can be converted into pluripotent stem cells by expression of certain transcription activators, a group of 4 transcription activators, I think, and they produce induced human pluripotent stem cells. These stem cells are introduced into the blastocyst that has defective or deficient in pancreas, giving pancreas development. So it will produce a chimeric blastocyst, and these human-induced pluripotent stem cells might occupy the region that will develop into pancreas, because that region would be deficient in the blastocyst embryo, and consequently a chimeric animal may be produced, is likely to be produced, that will have pancreas developed from human cells. And this pancreas then can be used for transfer into human beings, transplant into human beings, and this should be readily accepted. If the pluripotent cells were derived from a person uh, into whom the transplant is to be made, or a very close relative of this person. So this is something, a futuristic area of application of gene editing that could be that could mean billions of dollars to human health industries. Next one, please. So thank you. In the end, I hope I have been able to give you some idea of gene editing, basics of gene editing, and how it can be used for human welfare. Thank you.
Thank you